All right, we talked about mitosis and cell regulation and cancer, and now I'm just going to wrap this up with a real quick discussion about binary fission. So binary fission is how prokaryotes divide. Now, remember, prokaryotes have no nucleus or membrane-bound organelles. So that's good for us because then we don't have to worry about any of those. They do, however, have a cell wall. So most have a cell wall, so we got to deal with the cell wall. So this is going to be the same but a little bit different. So let's draw ourselves a bacterial cell. And bacterial cell DNA is usually in circles like that. Okay? So this is just going to be DNA. Okay? And then they do have these little, oh, I don't want to use the same color. Let's see. They do have these other little proteins. So they're not an organelle. This is a protein, and it's called FITs. And I don't necessarily care that you know what it's called, just that there's this little protein. So what's going to happen is, this is called an ORI, O-R-I, or origin of replication. I should write that out here. Origin of replication. And the DNA is going to replicate. And unlike our DNA, which can only go, it has to go in a certain way, in a certain direction, these guys are going to copy like this. So they're going to go from either side of the cell, basically, and then they're going to create a copy of DNA, of this same DNA. So here's 2 copies of circular DNA. All right? So next, easy enough. So next, let's take our guy here. And now our proteins, our DNA is going to start to split and go to either side. So you have one over there, one over here. And all of our little fits are going to start to line up in the middle. Kind of start hanging out in the middle. Okay? And then it is going to pinch this thing. Remember our cleavage furrow. All right? Just like when we are doing cytokinesis, we're going to have a cleavage furrow. And this is one. Okay. Where we have our DNA, of course, one over here, then we have our other one over here. And our little proteins are actually going to end up going down the middle right like this, called the septum. And so what's going to happen is that those proteins are actually going to create a situation, they're going to create a septum. So these proteins are just going to line up and create a septum. And this septum is going to divide until we get our 2 cells over here, and they're going to have their DNA in one, of course, but then they're also going to end up having their cell wall back around the outside of them. That was made with the septum. Okay? So usually bacteria have some kind of cell wall. So that's the only part that we really have to deal with, is getting this cell wall that's around it to form all the way around. Okay? And that's what we need those tricky little proteins for. And so what you end up with is 2 identical, genetically identical O-A-L-L-Y. Identical cells. All right? Oops. How did I make an eraser? Cells. All right? And that's it. Easy peasy, pumpkin squeezy.